Okay, so thank you, Jörg. So the floor is now open for questions, and I can see that there's a hand raised over here. The microphone's coming to you.
Okay, my name is Torbjörn Lundstedt and I'm from an SME, Acure Pharma, which are based on the concept of intelligent combinatorial chemistry. And I have one question here. I don't know if I misunderstood this, but you're very focused on the number of compounds, not anything about the quality of the library. Shouldn't it be any quality control included?
So, Jörg, I think.
Absolutely.
But the microphone
doesn't work. Yeah, no, it works. Absolutely. Quality is goes without saying. That's why it's not included here in these slides. I think industry has went through a sometimes painful process of discussing all these different aspects. And I think currently we have a common understanding what the quality standards are, and they will be implemented here for this aspect as well. Yeah? But it's also, in terms of finding the right partners, we need to be clear on the ambitious goals that we have. Yeah? That's why we put in the numbers, but quality is equally important.
But traditionally, in all the combinatorial libraries, I think it's been very good. The purity of the compounds has been very low.
Yep, that's why we are asking you to make it better.
I think as well that when you read the call text, it's one of the functions of the European lead factory will be to curate the compound collection and to assure compound quality as well. So it's a very important point, and it is considered within the call.
I mean, I agree with this statement that probably early days combinatorial chemistry libraries, they had their problems, no doubt about that. But also there we have seen a learning curve. Yeah? And build on that learning curve, we would try to be even better within this project.
Okay, so.
Ursula Teurelsbacher, Austria, Vienna. We just heard that one of the public benefits should be a starting point for orphan diseases, or let's say the less attractive, commercially attractive disease areas. So some of the disease areas require very specific compound libraries that have different criteria. One example is antibacterial drugs. And they also require different screening assays. So my question would be, how do you make sure that the result of these projects is expanded to the more neglected or less commercial interesting disease areas, and that it really covers more than we have seen already?
Yeah. The answer is pretty simple. It's going to be very difficult to purposefully design the processes towards orphan diseases. Yeah? We might generate synergies there, but we have to live, for example, with the compound library that has been introduced. We do not, or there is also no way, at least from that I can envision, to somehow bias the libraries that you can tackle antibacterial projects, for example, with a higher likelihood of producing results. What we can provide here is another opportunity. Yeah? You can create synergies, and that is it. Yeah? In the end, it's going to be tailored towards EFPIA. Yeah? And you can make use of that, what is going to be provided, but it's not intended to be this screening platform for orphan diseases. But I can see a lot of fields where we can create those synergies.
Waterloo, KU Leuven. So I had a few practical questions. One is about the mention of targets all throughout your presentation. Now the target-based throughput screening has come under some criticism recently, and the question is, do you also envisage phenotype-based screening? Secondly, you have mentioned that you want complementarity between the targets or the proposals from academia and the ongoing work in industry. This assumes that people know what the industry are doing, and are you prepared to make this information available to assure complementarity? Otherwise, people may be sending in a lot of proposals that are dead on arrival. I don't think it's very fair to people who put in all the work. And finally, if people just provide you with an albeit interesting target, who's going to do the assay development?
3 questions. Let me start with the last one. Who is going to do the assay development? Well, we have to see what the winning consortium will be, yeah? And they will clarify then the question, who is going to do the assay development. That's not within the presentation, that's not within the text. We don't know that at this point. We have to see what is the composition of the winning and selected consortium in the end. Phenotypic versus target-based screening. I agree, especially in the academic scene and also really with increasing relevance in industry research, we see a lot of phenotypic screening. Of course, that's also part of this initiative. We have to see how to handle that. It will be part of the negotiations and discussions towards the full project proposal, how we handle that in the EFPIA group together with the full consortium in the end. Providing ideas what industry is doing, that's certainly a very good point. We have to find a way to provide that information. Yeah, that's pretty clear. I mean, many of these pieces and bits of information are out there, of course. For the interested people, it's fairly straightforward, I think, to get a good picture what the project portfolios of individual pharma companies might look like. Yeah? But I agree, we have to be fair to everyone who is applying there. We have to give a good idea what the intention of the consortium is, what the focus is. We will make a positive selection later on, of course, on those which are complementary.
Okay, so we have 2 questions down here, and then there's a cluster over on this side. So first,
Phil Jones, independent consultant. I guess I'd like a few comments on your thoughts on sustainability. The original proposal talks about what will happen after the 5 years, and I'd like to hear your comments on that. And in particular, is it envisaged that the EFPIA compounds that are part of the collection, anything that's left will remain as part of the collection after that time?
Yeah. Also, to start with the last comment, yes, that's true. Everything that is left over after the 5 years of the EFPIA compounds will stay there. Yeah, but the numbers are typically in that way that, on average, it's very likely that there is not much left. They will be consumed during the process. Yeah. The first question was
about the sustainability and what your thoughts on what should remain.
Sustainability is, of course, a very difficult issue. We do expect, of course, also from the applicant consortia to bring forward ideas what their concept might be. They will look different, those concepts, if you have different consortia. But since we are also here in that business of generating starting points for drug discovery programs, joint IP, there is also value in there, and that's a focus of this project. And the value that you generate will be the basis then for any model on sustainability here. Yeah. That's the starting point, at least. This is also something that needs to be clarified during the preparation of a full project proposal. It doesn't make sense, and I agree with that completely, to grow something here over a 5-year funding period where you have no idea how to sustain those 30, 40 people maybe in a screening center after the funding period. That's pretty clear.